Our previous videos demonstrated procedures for imaging bioluminescent and fluorescent sources in vivo in 2 dimensions. With these methodologies, we collect the light that reaches the surface of the animal from various locations within the body and calculate cumulative surface intensity. Although 2D imaging is highly useful for tracking intensity as a function of time within a region, particularly in response to a stimulus or treatment, we cannot discern the absolute amount of signal because tissue attenuation is not accounted for. Therefore, diffusion patterns are not entirely representative of the location, and certainly give us no information concerning depth of the sources in the animal. 3D tomography helps further that biological characterization by allowing pinpoint localization of your probe within the animal. Tomography also allows for quantification of a fluorescent agent's accumulation within the tumor or intended target in absolute concentration units. In addition to our 3D optical reconstructions, we will also be performing micro-CT imaging to co-register functional and molecular optical readouts with anatomical high-resolution micro-CT data. The Revity IVUS Spectrum CT will be used for this video as both 3D tomography and integrated micro-CT are included standard features. We will walk you through the procedure for capturing and analyzing your 3D optical image. Before beginning, perform a kinetic analysis to see when emission is highest and reaches a plateau. This is the time post-injection when you will acquire your images. We perform our calculation assuming a consistent light output, and therefore, it is critical that the data be acquired where light output is most consistent, specifically in the plateau. To perform bioluminescence tomography, also referred to as diffuse light imaging tomography or D-light, we take advantage of the broad emission spectrum of luciferase as it overlaps the hemoglobin absorption curve. Essentially, we collect signal that reaches the surface of the subject using a range of spectral filters corresponding to the firefly luciferase emission peak from 560 to 640 nanometers spaced every 20 nanometers. This allows us to generate and solve a set of linear equations to reconstruct the depth and determine the total flux of the light source in the body based on the amount of attenuation occurring at each wavelength. This can be determined based on the intensity and shape of the resulting curve. Let's begin by setting up the acquisition software. In the IVUS System Control Panel, start setup by clicking Imaging Wizard and selecting Bioluminescence, D-Light, and then the reporter you're using for your study. We will select Firefly. Click Next. As demonstrated previously, complete the last window to reflect your setup and acquisition parameters. Select the CT resolution that is desired for the acquisition. We streamline CT settings to maximize resolution and throughput, while minimizing damaging exposure to radiation. Therefore, it is only necessary to choose the desired resolution for your scan. Current and voltage will be automatically selected for the chosen setting. At a minimum, we must capture a fast scan of the animal to generate a surface which will be used in the optical tomography rendering. You have 5 choices: fast, standard, 2-mouse standard, which is bioluminescence only, medium resolution, the best resolution for soft tissue contrast, and high resolution. Please be aware that the high resolution setting is for CT only and cannot be combined with optical reconstructions. CT resolution can be selected from this drop-down menu. We will utilize the default setting of 1-mouse standard. Click Next. The sequence editor will open and each filter pair selected will represent one image in the sequence. Click Acquire Sequence. Label the images appropriately. Once the instrument has finished acquiring the images, the status light on the instrument will change to green. CT reconstruction has begun in the background during the optical image capture, and you should see the reconstructed CT scan open in a separate tab in your sequence window. At the end of the acquisition, we should ensure that each image has between 60 and 60,000 counts per pixel. We should strive to attain 600 counts for each image in the sequence, however. This is often difficult using filters below 600 nanometers. Try to obtain this level with the 600, 620, and 640 filters if possible. Also reacquire at lower sensitivity settings if saturation occurs. We cannot analyze saturated data appropriately. We will now continue to fluorescence tomography or flight acquisition. For this study, we will overlay the D-light and flight optical reconstructions. If this is desired, you cannot disturb the animal between acquisition of these 2 sequences as the overlay is a direct copy-paste procedure. If the animal is disturbed, the sources will not align appropriately. Our objective with 3D imaging is twofold. Firstly, to estimate the location of these fluorescent signals, and thereby validate agent colocalization within these lung tumors. Secondly, to estimate their absolute concentration in picomolar units. Flight analysis utilizes a transillumination light source to project excitation light from below the animal through the tissue. The same excitation and emission filter pair will be used for every image in the acquisition. However, the position of the transillumination light source will change for each image. It is this position relative to the surface intensity pattern, and changes in those patterns as the transilluminator moves, that facilitate the analytical procedures to elucidate location within the animal. As with D-light, tissue attenuation and diffusion are accounted for in the reconstruction process. Thus, output can also be provided as absolute intensity in picomoles, or as number of cells. For this experiment, we injected the mouse with the probe 24 hours prior to imaging. We may now set up the software for fluorescence tomography. In the IVA system control panel, click on the imaging wizard, select fluorescence, flight, then choose your reporter. For this study, we are using IVASense integrin receptor probe 750. If your reporter is not in the alphabetical list, you can manually enter in the excitation and emission wavelengths. Click Next. Complete the last window of the wizard to choose your imaging subject, acquisition parameters, auto or manual, and field of view. Select the CT resolution. We will be using the standard one mouse option. Select Transillumination Setup. In the Transillumination Setup window, select a minimum of 8 to 10 points immediately surrounding the source of interest. Here we are interested in the thoracic cavity. If you're uncertain about the bounds of your target or the exact location, you can perform a quick 2D raster spatial scan of the entire animal. For this model, we are looking for the fluorescent source in the lungs, so points will be chosen in and around the thoracic cavity. Timing can be as fast as a few seconds or as slow as a couple of minutes per point selected. Select points covering your region of interest, but do not select the entire animal. This will help reduce scan time. Select OK. Then select Next in the imaging wizard. The sequence editor will open, and each point selected will represent one image in the sequence. Click Acquire Sequence. Label the images appropriately. Once the instrument has finished acquiring the images, the status light on the instrument will change to green. CT reconstruction has begun in the background during optical image capture, and you should see the reconstructed CT scan open in a separate tab in your sequence window. After flight sequence acquisition is complete, do not forget to remove the animals from the stage and return them to their cages. Note the two tabs at the top of the image window. The left tab contains the individual fluorescence images taken at each transillumination point selected, and the right tab shows the 3D view including the CT scan and the reconstructed optical signal. Now that both flight and D-light acquisitions are complete, let's analyze the data we've collected. The first step in analyzing any 3D optical data is to generate the surface topography that will define the area within which our bioluminescence, our fluorescence signal, will be reconstructed. Please note that the following directions are specifically related to the IVAS Spectrum CT and Spectrum CT2 instruments. For Spectrum and Spectrum 2 instruments, surface topography generation is slightly different. Follow the steps in our technical note or user manuals, or contact a Revity representative for assistance. To generate a surface topography on a Spectrum CT, go to the Surface Topography tab on the tool palette. A predetermined threshold defining the boundary between tissue and air or noise is set by the software as a default. Click Show to see this threshold. Although the threshold slider can be used to adjust this, default settings are usually optimal. Best practice is to reconstruct first with default, and if the generated surface is not acceptable, you can return and adjust for a more accurate boundary. You should save the results of your surface generation by typing a name in the Name field. Let's name this D-light surface. Then click Save. Now that we have a surface created and saved for our animal, the next step is to reconstruct our bioluminescent source. Close the Surface Topography tab and open the D-light 3D Reconstruction tab. Under Analyze, you'll notice that the various emission filters that were used at acquisition are highlighted and checked. The number next to the filters shows the applied threshold percentage, which will correspond to a minimum photon count number that is slightly more than the typical noise floor for a bioluminescent acquisition. In other words, the data is threshold just above a typical noise floor, so that we exclude noise but include the majority of light that makes up the diffusion pattern. Under Properties, you will see the tissue properties set as mouse tissue and source spectrum as Firefly. If the wizard was completed appropriately, these selections should be showing as default, but confirm your tissue type and source type before proceeding. To proceed, click on the Start button in the lower right corner of the Analyze subtab. This will open a data preview window where you can view the individual images that were acquired with the specific emission filters at the applied threshold. Although you can change thresholding, it is recommended to first proceed with default settings. A subsequent reconstruction can be performed with custom settings if you wish. You can change the default thresholding by clicking on the Data Adjustment button, after selecting an image, or simply by double-clicking on each individual image, and then adjusting the threshold adjustment slider within the data adjustment window that appears. Keep in mind that while there is no one particular threshold percent that works for every model and experiment, Some amount of thresholding is usually helpful in getting a more accurate reconstruction of the bioluminescent source by excluding noise. A good rule of thumb is to adjust the threshold slider so that the minimum count number is greater than 100 counts. In this same window, you can also use the region selection tools to mask in a particular region or regions to be used in a reconstruction. When finished, click OK to close the window. After your thresholding and masking are completed, click the Reconstruct button to launch the 3D reconstruction process. The result will appear within the topographic and CT volume as a volumetric color map. The results should be saved by typing a name in the name field and clicking Save. The 3D bioluminescent volume is automatically overlaid with the skeletal detail, and the full rendering appears in the 3D View tab. This tab consists of the active viewing window on the right, along with the various anatomical slice planes on the left: coronal, sagittal, and transaxial. You can toggle the slice planes on or off by clicking the red, green, blue plane icon in the toolbar above the active viewing window. You can also rotate the entire 3D image by holding down the left mouse button and dragging in the direction you wish to move the image. Take some time to get familiar with manipulating the 3D image. If you lose your orientation, click on the Reset View button to go back to the standard top-down viewpoint. Visualization of the reconstruction can be adjusted using the 3D Optical Tools tab. You can adjust the surface, the optical source, or co-register the result with a mouse atlas. For more information on these tools, please see the user manual or contact a Revity representative for help. 3D optical data can be analyzed somewhat similarly to 2D data by drawing ROIs and reporting a total flux readout. Later, we'll show you how to report out a cell number based on the titration of cells that we did earlier in our study. When doing ROI analyses with 3D data, we need to use a 3D ROI to capture the volume of interest. To do this, expand the ROI Tools tab on the tool palette and click on the 3D ROI icon. This will place a cube ROI on the image. To position and manipulate that ROI, click on the 3D ROI Transform tool icon and select the ROI. In this case, ROI 1. With the ROI now selected, you can transform the ROI 3 different ways. First, you can position the ROI in the X, Y, and Z axis. Second, you can grow or shrink the ROI. And third, you can rotate the ROI. Pressing the Tab button on the keyboard allows you to alternate between these 3 different transformation tools. When you're satisfied with your ROI, click the 3D ROI Transform tool icon once again, and the ROI will be locked. To see the total flux readout for the ROI you just drew, click on the Measure 3D ROIs button, and then make sure that the 3D ROI Measurements tab is selected. Choose Source Voxels from the Data Types pull-down option. Also, make sure that photons per second is selected as the measurements unit. Data can be copied and are exported as previously described. If you prefer to see a readout of cell number instead of total flux, we will need to create a quantification database derived from a serial dilution of luminescent cells in a well plate. We serially diluted cells at a 1:2 dilution with a starting concentration of 500,000 cells in a black well plate and imaged the plate. To create the database, we will load the image and then go to Tools, Well Plate Quantification in the toolbar. We will want to utilize wells with signal above 600 counts as shown previously in the in vitro bioluminescence video. After determining the wells you wish to utilize, then choose the well plate type and the pull-down option list in the upper left corner. In this case, we'll choose 12x8 for our 96 well plate. Then position your grid ROI as we've shown previously. In the well plate quantification window, populate the wells with the dilution values. Select those wells and click Set in the Sample Wells row when done. If you have background wells, select and set those in a similar fashion. After sample and background wells are set, click on Quantify in the lower right-hand corner of your screen. The photons per second per cell results will be shown. Before saving, confirm the linear fit of the data and the accuracy of the dilution by clicking Quantification Plots. There should be a strong linear correlation between well plate population and total flux. You can generate and save this database to the instrument you're working on by typing in a database name and clicking Save on the bottom right-hand corner of the window. If you wish to transfer the result at a later date, then save the result in the left-hand corner of the window as well. This saves a copy to the well plate image file and allows you to copy and transfer the result, if desired. Now that we have generated the database for our cell line, we can go back to the 3D view of our D-Light dataset. Under the D-Light 3D Reconstruction tab in the Tool palette, click on the Properties subtab, then select the luminescent calibration database that we just created. You will notice that your scale bar will be represented in cells now, and you can also display total number of viable cells in the ROI Measurements window. Now let's open the 3D fluorescence data that was acquired. First, use the browser tool to load the 3D fluorescence dataset. Next, just like we did with our D-Light dataset, create a surface topography. Let's name this one flight surface and save it. But remember, since we didn't move the animal between our bioluminescence acquisition and our fluorescence acquisition, the surfaces for these two datasets should be nearly identical. Later, we will show you how to overlay the D-light and flight optical reconstruction so that we can see bioluminescent signal and fluorescent signal at the same time in one animal. For now, let's finalize the fluorescence optical reconstruction by opening the Flight 3D Reconstruction tab on the tool palette and clicking on the Start button within the Analyze subtab. As with the previously analyzed D-light dataset, you will see three tabs: Analyze, Properties, and Results. Under Analyze, you will see each image in the sequence corresponding to the number of transillumination points that were chosen. The excitation and emission wavelengths, as well as the predetermined threshold of each image, is shown as well. Follow the guidelines previously discussed for thresholding if needed. Press Reconstruct to begin the process of reconstructing your fluorescent source. The completed flight data can be analyzed as previously described. 3D ROIs can be drawn around an area of interest, and the ROI cube can be positioned, stretched, and rotated. Once you're satisfied with the location and adjustment of your ROI, you can report total fluorescent yield by clicking on the Measure 3D ROIs button and selecting the 3D ROI Measurements tab. Make sure Source Voxels is chosen from the Data Types pull-down menu, and the unit beginning with picomoles is chosen as the measurements unit. The process for creating a fluorescent quantification database is similar to the process for creating a cell count quantification database. Instead of cell number, you will enter the nanomolar concentration and volume per well. In our example, we started with 100 microliters of a 20 micromolar solution. A 1:2 dilution series was performed in a black well plate. After pressing Quantify, the extinction coefficient and cross-section are calculated to facilitate the conversion of our total fluorescent yield unit to picomoles of compound. After applying the database to your dataset, the scale bar will present in units of picomoles, as will the measurements table, if picomoles is selected. Even though we acquire bioluminescence and fluorescence information from the same animal, so far, we've only looked at the corresponding D-light data and flight data as separate datasets. But what if we want to look at the data together at once? To do this, the first step is to make sure that both the D-light and flight datasets have been loaded and that the reconstructions are complete. Next, change the color of the voxels for each data set to a separate, distinguishable color. Here we'll make our flight data red by opening the 3D Optical Tools tab in the tool palette. We select the Source subtab and choose Red from the Color Table pull-down. The same process will be followed for changing the D-light data voxels to green. With the D-light data and flight data now easily distinguishable by color, open the flight dataset window, click on the Copy-Paste button, and select Copy Source Voxels. The flight data has now been copied to the clipboard. Now open the D-light dataset window, click on the Copy-Paste button and select Paste Source Results. At this point, you'll see D-light data and flight data, D-light in green and flight in red, together in the same animal. If you would like to see areas of colocalization, go to the Tool palette, 3D Optical Tools tab, Source tab, and check the Maximum Intensity Projection checkbox. Overlapping areas of red and green will show as the blended color, yellow. Overlaid sources are also visible in the Slice Planes view on the left-hand side of the window. To finish up your 3D reconstruction, the CT rendering can be adjusted to create the visualization you desire. Open the 3D Multimodality Tools tab in the Tool palette. We will highlight a few tools that may be helpful to you. The Volume subtab houses most of the tools you would need to adjust visualization of your CT reconstructions. In this tab, you will see a logarithmic histogram of the density of all the voxels that make up the CT image. The unit to describe the radio density of an object is called Hounsfield units. The CT image voxels display as a grayscale image. Low-density objects like air will appear darker and have a lower Hounsfield unit value. High-density objects like bone will appear lighter and have a higher Hounsfield unit value. On the far left of the histogram are voxels of low density with lower Hounsfield units. And on the far right are voxels with the highest density corresponding to higher Hounsfield units. The default view is a blended view showing some low-density voxels and some high-density voxels. In this study, we only wish to show the skeleton, so let's start by clicking the AutoFit Air Noise Boundary button. This effectively takes a slice from the histogram that shows the skeleton of the animal bounded with the surface of the animal. Now grab the boundary bar and slowly slide it to the right until only the skeleton is visible. You can double-click on the top of the bar to adjust the color of the voxels above the threshold. You can also crop the image and adjust the opacity with the provided slider. Under certain conditions, even more advanced organ segmentation can be done using the CT data. For more information on how to do this, please contact one of our advanced imaging training specialists. Now that we have the combined optical image with the desired CT adjustments, We can export a static image of the 3D data. Position the animal as desired in the image viewing window, then click the Export Graphics button. Give the file a name and select any of the standard file formats, or just leave the file in the default JPEG format. Alternatively, you can copy the image directly into PowerPoint for presentation. An even more compelling way to show 3D image data is to create a video animation. To do this, click on the Tools button on the Living Image menu bar. Then click on 3D Animation. A separate 3D animation window will pop up where you can choose from a number of preset animations. Choose the preset animation you wish to use from the Presets drop-down menu. Here we'll choose Spin Clockwise on the Y-axis. You can click play at any time to see video. Additionally, you can create your own custom views or call out particular areas of the animal by creating a custom frame. For instance, here we will zoom in on the chest and select the Add New Keyframe tab. The software will interpolate how the image should move between the final frame of the preset and the zoomed view. A final smooth animation will be produced when you complete the video. To export the video, click Record to save the animation in one of 4 standard movie file formats. In summary, we have demonstrated the utility and benefits of 3D bioluminescence tomography to identify the location of tumors in the lung and quantify the absolute intensity of sources as a function of viable cells. Furthermore, 3D fluorescence tomography was incorporated to validate accumulation and targeting of these tumor sites by our fluorescent agents and to estimate agent concentration. We've utilized 3D imaging to answer pertinent questions concerning the experimental model that were not possible by simple 2D analyses. We hope you take advantage of these powerful methodologies to supplement your current 2D work. Thank you for watching.